So thank you for setting this up. Thank you for inviting me. It's always a pleasure to share what we have done. And today I will focus on the immunoglobulin G and its role as a functional effector of aging, diseases, and mortality, and also show some of the data which are still unpublished. And just a short introduction. I believe everybody here knows how important glycans are. This is the example I usually use to try to convince people that they're important, saying that, this is a glycoprotein. Everything here is a glycan. Red and white dots are protein amino acids. And if somebody is focusing only on a polypeptide part or a gene for a specific protein, it's like they're studying a bird without a single feather. And we can be the best scientists in the world, have all the money, have perfect experiments. But if we are trying to understand birds and how they fly, and we use the model without its feathers, this is the closest to the flight we could come, the bird walking. And the same goes for proteins. For example, immunoglobulin G is one of the key weapons of our immune system. But what will this antibody actually do depends on the glycan on the FC domain. And this is not encoded in a gene. It's an interaction of dozens of genes. And of course, to understand full function of proteins, we have to look into glycans. And you all know that glycation is something completely different. Glycation is the chemical damage of glucose, like taking a shotgun and firing at the proteins. Glycosylation is additional layer of complexity. So the polypeptide part is encoded by a gene. This is the evolutionary wisdom from billions of years. We don't want to change it. And we are 99.9 identical in our protein coding sequences. Glycan part is where we differ. It's encoded in the network of genes. This is the network of genes we mapped for the IgG glycome, and this then determines the entire structure. So from the functional point of view, alternative glycosylation, putting different glycans to the same protein, is like a coding mutation. And something I keep asking, and I haven't heard it in the past few presentations, is if you talk about glycoRNA, is anybody looking at alternative glycosylation, meaning different glycans attached to the same glycoRNA? This would be an interesting thing to look into. And if we talk about the evolutional or informational point of view, then the glycome composition is a complex trait, meaning we can be different without the risk of our genetic heritage. And in this special issue of engineering, we are bringing a paper where we are finally providing some evidence that it was actually glycosylation which enabled the appearance of multicellular life, because by doing phylostratification, we showed that the peak of evolution of protein glycosylation was actually happening when the life was becoming multicellular, when we started to form cell colonies, multicellularity, and so on. So glycosylation itself is ancient. It comes from archaea, but the glycoprotein evolution peaked when the life was becoming multicellular and getting all the diversity it has today. And this is also what we see in a human population. So if we look only at the protein part or the protein coding genes, we are more or less all the same. If we look at glycans, we are very different. This is a glycome composition, IgG glycome composition of 6 random individuals. Clearly, people are different. And unfortunately, the big majority of scientists were focusing on genes for the last 5 decades. And despite the technical success of genome analysis, we can now sequence a genome for less than 200 euros. There has been very little translation, and actually it's a big disappointment for everybody. And I think this will never change, because only about 30% of complex diseases is genetic, 20% of longevity is genetic, even less. Some studies say 6%. So we will never understand longevity and diseases by looking into the genome. And if we talk about the medicine, personalized medicine is still a dream. We think we have a modern medicine, we don't, because these are the best money-making drugs in the pharmaceutical industry today. And the best of them you have to give to 4 people for one to benefit. You're actually harming 3 persons for one to benefit. And this is the best drug. some of the drugs you have to give to over 20 patients for one to actually benefit. And this is a Nature paper from a couple of years ago. And to move from where we are today into the modern medicine of future, we need biomarkers. We need tools which stratify people and identify which people respond to a specific therapy. And I think for that, we need glycans because, okay, sorry, what I want to say, so this is a modern medicine. If we talk about the lifestyle, the traditional medicine or whatever, we are flying blindly. We don't have good tools to look into each individual patient. And I think we should go into glycans because glycans are the best biomarker in personalized medicine. The first one ever introduced and still probably the most successful one, the blood groups, A, B, and O blood groups. These are glycan structures. So you give a transfusion of a wrong blood group, you are killing a person. So you are saving millions of people by doing the glycan profiling for them before any surgery. But unfortunately, the majority of the glycans are not being investigated simply because they're complicated. There are many different structures which can be attached to the given glycosylation site, many different sites on the same protein. You need many steps to get reliable quantity data, and not so many labs do it in a good way. So there was a comparison study done by the National Institute of Science and Technology in US a couple of years ago. They checked 76 labs with 103 different data sets generated, and unfortunately, the results were all around the board. Some of the labs had CVs of something like 100%, which is horrible. And it's not easy to work with glycans in high throughput. So this is one example of our studies. So each bar are standards on a different 96-well plate, and we are just doing it routinely. And then you see something happen. There is something like a 50% increase in these glycans, increase in other glycans. And this is the standard, this is the same glycan. So this is not biology. This is our analytical imperfection. And if you don't have enough standards and you don't have technology which is looking for that, then you will report this as biology. If you're using standards, then you see this is an aberration, you repeat it or you fix it, and then you have better quality data. And this is what we are focusing on for the last practically 3 decades. Professor Wang mentioned one of the grants we had. We have multiple European grants. We are currently running 15 Horizon grants in parallel. We got more than 40 million euros, which enabled us to develop technology which is really working well today. And a recent review in a chemical reviews identified that out of the 191,000 glycomes published by 1921, I believe, by 2021, my lab generated 159,000 of them, so over 80% of the global output. And we did it with collaboration with some of the best researchers all around the world, generating data which can be used to understand human biology. And today, we do have a technology which is high throughput, which is clinic ready, and which I hope will be in the clinic soon. We have a dedicated glycan age profiler made actually by Nanjing Super Years. We are developing dedicated kits which are CE-IVDR compliant, meaning they do have a regulatory approval. And then this generates results with the CVs which are really acceptable, and you can do proper clinical work with them. Something we already have on the market is something called the glycan age test of biological age. This is a test where you have a home kit for sampling, get a dried blood stain, ship it to the lab quantifies IgG glycome, and then determine effects of different lifestyle factors on your future health. And of course, I'm an inventor and a patent. I'm a co-founder of a company. I'm here heavily conflicted, so you should not be taking my word for that. You should look at the data and make your own conclusions. But if you look, for example, how IgG glycome changes through age as we are getting older, and this is a study on over 6,000 people from China, Croatia, Estonia, Scotland, England, you see a very similar trend. Men moving linearly, women being younger in the reproductive phase, then rapid acceleration, and then catching up with men. And this kind of replicates through cohorts. But the most important thing is, this This is a standard human. So standard human does not exist. Nobody is standard. We are all individual dots somewhere in this cloud. An important thing for each of us is, are we below or above this kind of average rate of aging? And this depends on our lifestyle. So healthy lifestyle makes you age slower, and unhealthy makes you age faster. But to be able to see it, you have to monitor an individual. And this is what technology we have today enables. What you see here, this is my personal IgG glycome composition, something we published in Neuroscience 2 years ago with Mike Steiner from Stanford. And what you see here is these numbers do not change unless there is a biological change. So analytically, we are really good. But then something can happen and the numbers deviate. Although the change seems to be small, but it's significant. There is a real change here. And these were the glycans which are pro-inflammatory going up, the one protecting from heart attack and stroke going down. So something bad was going on in me. And the regular blood work didn't show anything. And actually, I spent quite some time to find that I actually had a cryptic root canal infection, meaning I had bacteria growing in my root canal, which is known to be a risk factor for a number of different things. When I fixed it, the biomarkers went back into the normal direction. So you can actually now today using this technology monitor an individual in a reliable way. The question is, does it mean anything? So does IgG glycans predict anything? And this is pretty much exclusive. I haven't talked about it yet. We just posted it on meta-archive actually last week. This study on 5,000 people collected in Germany 10, 15 years ago. This is their age. This is their glycan age. And the blue ones were still alive 10 years later. The red ones died in the meantime. Meaning, if you have a higher glycan age, your risk of dying is much higher. And it's actually 5% higher mortality risk in this cohort for each increase in the glycan age year. Of course, to claim something like this, it cannot be done on a single cohort. So, of course, we did the replication cohorts, 2 of them actually, one in Karolinska, 1,000 twins followed through 30 years. Another one in Generation Scotland, again, the same glycans, very similar hazard ratios. And then we did another replication study with Peter Joshi in Edinburgh again. So we can pretty confidently say IgG glycome composition predicts risk of future all-cause mortality. And it also correlates with a number of different diseases. So in this same paper we posted last week, we have over 20,000 people, I believe around 30 different diseases. And this is the effect of aging. And more or less all diseases we looked into, from diabetes to inflammatory, cancer, and so on, people have higher glycan age if they're ill than they are sick. So these glycans are not good biomarkers for any of the diseases. But if you look the other way around, they're actually a very good biomarker of resilience, meaning how resilient you still are for developing the disease in the future. And we see these changes in glycans up to a decade before people get diagnosed with the disease. There are many prominent papers. I don't have time to go through them. And then again, something exclusive, unpublished yet. This is a large cohort, people who are completely healthy, and their IgG glycome, this index of health. These are people who are still mostly healthy, but you can see something is going wrong, and you see many of them already change the glycans. And once they're diagnosed, majority of them actually have the change in glycan. So glycans are very early predictors that something is going wrong, and this is not surprising because glycans, IgG glycans, have so many different roles in balancing inflammation. And we know that inflammation is so important for future disease development. And I'll show you one example what we have done in the cardiovascular disease space. So the first paper was published now 8 years ago showing correlation between the disease risk and glycans. Then we got samples collected 30 years ago where actual people, we had samples 30 years ago, had a heart attack or stroke in the follow-up period. And the IgG glycome composition in men was as predictive as the entire AHA score. In women, even a bit more predictive. But most interestingly, different glycans were predicting heart attack and stroke in men and women, meaning we have a sex-specific role of glycans in CVD development. Published well, but again, nobody trusts a single paper. We did the replication cohort. Again, the same glycans, very similar hazard ratios, separate in men and women. We did additional 2 replication cohorts. This was published 2 years ago in a Circulation Research, and we even moved to the different parts of the world. And this is a study we did in the Middle East, showing that the same glycans also predict this premature myocardial infarction, which is a big problem there. The best part of it is maybe there's a way to fix it. For now, it's only mice. But if you feed mice with a MANAC, which is N-acetylmanosamine, which is a precursor for salicylic acid, you can protect them for hypertension even when they develop obesity on high-fat diet. But of course, for now, this is only on mice. And what we really want to learn is there something we can do in people? Can we revert this glycan age clock of aging? Can we slow down inflammation and be healthier for longer? We did a number of studies showing that there are things which work. I don't have time to go through all of them. I'll just show you a few examples. For example, caloric restriction works. Probably because we all eat too much, but we had 1,000 people on 800 calories on a day for 2 months. They got better. It works even on a prolonged period. So these are people who were on a low-calorie diet for 2 years. We published this together with Luigi Ferrucci, who is the director of NIH Institute of Aging. But the moment you try something else, and this is 5 different healthy diets, some people got better, some people got worse. It's just, there's no standard human. We are all different. And there will never be an intervention which works for everybody. At least we haven't found it yet. But the good thing is losing weight helps. It helps in your 30s, in your 40s, in the 50s. So obesity is something which can be fixed. There are some magic pills. If you want to reduce inflammatory potential of IgG, you can take things like infliximab. But that's a monoclonal anti-TNF. If you do it, eventually you will get cancer. So that's not the way to do it. But even infliximab didn't work in everybody. There are some people who did not respond. So we are all not the same. We cannot take the same drug. Estrogen is the other thing which works very efficiently in changing IgG glycome. We had a cohort of people with the gonadal hormone completely being blocked chemically, then either supplemented with estrogen or placebo, women on placebo aged for 9 years in a few months. The same happened in men, but in men, it's not testosterone, which is the active component, it's actually estrogen. So if you block conversion of estrogen to testosterone to estrogen, it doesn't work. We see this also in human populations. So the woman kind of age slowly before the menopause. In perimenopause period, they age rapidly, and then they catch up with men. And in a clinical work on individuals, so for this is one client of glycan age, very good glycan age, aged 30 years within a couple of months, first symptom of perimenopause, and then fixed with the hormone replacement. So IgG glycome can actually be used as a first indicator that we are losing estrogen, because this is not easy to quantify in perimenopause because it goes up and down within the day. We also recently published a first study showing that actually HRT works, again, in majority of women, and some women did not respond, so they had to see what's the problem here. There are some other things which work, like the therapeutic plasma exchange. Removing bad things from your blood somehow makes you younger. There seems to be some kind of a magic interventions, like isolating exosome fraction from the newborn pigs. You give it to rats, the rats live 50% longer. There are many drugs on the market which people sell for longevity, but most of them don't work. This is an example of metformin. We studied a proper placebo-controlled randomized cohort of people on metformin and testosterone. Testosterone did have an effect, but metformin helped only a single individual out of the entire cohort. These are the young healthy individuals, but if you take diabetics, of course, majority of diabetics will respond to metformin, not everybody. But the same thing is for SGL2 and GLP-1 agonists. So people are different, not everybody responds to the same drug. So the general idea is, and I'm finishing with a slide, is that we are healthy at the beginning, and then when one day we wake up with a chronic disease. And of course, did not happen overnight. This happened over a decade or even more of molecular changes moving from health to the disease. Usually, if we are diagnosed with a disease, this is beyond the point of no return. chronic diseases are not cured. This is why we call them chronic. But if you find people here with intervention, pharmacological or lifestyle, maybe we can postpone diseases, be healthier for longer. I'll just show you one example. This is my daughter Nicolina, CEO of Glycanage. She has a good glycanage, and then something happens. She immediately has a bad glycanage, but she's measuring, so she knows it. She can identify a problem and fix it, and then it's good again. And then maybe there's another problem. These are real data. So there was another problem, and then she fixed it again. So the idea is we don't wait for our tires to explode on a highway to change a tire on a car, but we wait to become sick before we do something. We shouldn't be doing that. Like changing a tire on a car, we should be changing things which don't work in our body, and in this way stay healthy for longer. And I thank all the funding agencies, and of course my team of people. This is the research team in Ganos and the translation team in Glycanage, and I thank you all for your attention.